So I'm in the nucleus. I'm copying my DNA. What do we call that? Transcribing. Expose the gene segment that I want to copy, unwind it, unzip it, expose it, copy it, make my pre-mRNA strand. Put a cap on the 5' end, put a poly A tail on the other end, the 3' end, so it knows where to start and it knows how to get the heck out of the nucleus. Got to get rid of those introns. Spliceosomes come in there, cut them out, splice them, the two ends together. Ta-da! I am now mRNA and I am ready to party. I leave through that nuclear pore right there, and I go out in the cytoplasm. I'm in the cytoplasm, but I am specifically hooking up with somebody when I get out there. If you know what I mean. I am hooking up with the ribosome. Okay, ribosomes in cytoplasm. All right. Because what do ribosomes do? They are involved with protein synthesis. That's something we looked about way at the beginning of the semester. Here we get to put it to use. All right. So why is this mRNA strand hooking up with the ribosome? Because that is where we're going to have the reading of our transcript of our mRNA. This reads it and translates it. This happens at the ribosome. And remember, it has two segments, right? It has a small subunit and a large subunit. Those two units come together when the mRNA strand comes out. And that mRNA strand just reads through it from the 5' end all the way to the 3' end. And we make the protein. Okay. We make the protein. So translation is when we read what we've written down, what we've transcribed, and we make a protein. That is what translation ultimately does. Okay. Reads the amino acids and puts it into a polypeptide. What's a polypeptide? Protein. Okay. Protein. All right. And again, all that happens in the cytoplasm, specifically at a ribosome. I have messenger RNA that's being made within the nucleus, coming on out into the cytoplasm. I also have tRNA, transfer RNA. Transfer RNA is what brings these specific amino acids to the ribosome. tRNA is what brings the individual amino acids, the specific amino acids, to the ribosome. I don't really care about the wobble hypothesis. Okay. Let's pretend that doesn't even exist. I thought I got rid of that. I guess not. Okay. We have a specific code. Okay. Here's that mRNA. Okay. And here is my specific code, right? Specific code. Specific code. We looked at that the other day, right? That genetic code. Those 3 little letters give me a specific amino acid. These letters that are on the mRNA molecule are what we call the codon. The tRNA molecule is able to read that and will bind to it with an anticodon. So who contains the codon? mRNA. Who contains the anticodon? tRNA. Okay, it's shaped like a T, right? On the exam, could I ask to give me the codon or anticodon? Yeah, I think I put that down as a potential question on the exam. So what does that mean? That means you have to know that C always goes with G and A always goes with U, or U always goes with A. Okay, that's what that means. So this tRNA molecule reads the codon, reads the message by pairing up the anticodon with the codon, and ta-da, I can bring the very specific amino acid for that specific 3-letter segment. All right. So what is translation? mRNA associates with RNA. And remember, RNA is ribosome. Okay. So you can just say ribosome. I'd be perfectly content and happy. You can call it rRNA as well, because technically that's what it is. Okay. The ribosome reads the information. It reads that codon. It reads that message that we just transcribed in the nucleus. The ribosome directs various different tRNA molecules to bring specific amino acids to the party. We have 64 different possible combinations. Each one has a specific amino acid that it brings. The specific code that the tRNA molecule carries is the anticodon. And again, it is reading the words, the code, the codon found on the mRNA molecule. Oops, sorry. If I look more closely, at the ribosome itself, it's actually really complex. It's not just two turds sitting on top of each other here, boom and boom like that. When you look at that ribosome, it's actually pretty complex. And here it is. There's a small and there's a large subunit, and the molecule of mRNA just gets fed right in between them. And that's what you're seeing, right there. Ooh, and it's purple too, see? Synergy. Okay. Okay, so it gets fed right in between the two subunits, and that's how we read this sucker. But when you look on the inside, you can see it has these 3 binding spots. The EPA. E stands for exit, P stands for peptide, A stands for amino acid. The A site, this is where you can write down if you'd like, okay? The A site, sorry about that, is where amino acid first attaches. Where amino acid first attaches. First attaches. ATTACHES, right? The P site is where we form the peptide bond, right? Because what connects two amino acids together? Peptide bonds. Again, this is stuff that we learned way back, chapter 5, I think. I don't even remember. It was so long ago. I think in the first unit. Where peptide bond, P-P-T-I-D-E, peptide bond forms between amino acids. And then E is the exit. This is where the amino acid leaves, detaches. I'll put detaches. Detaches. Okay. So there's actually quite a bit of stuff going on there, right? It's pretty complex. Okay. So here's I'm just going to start here and I'm just going to go this way with it, all right? So here is an amino acid. Okay. Here's my codon right there, right? Here's the tRNA molecule with the anticodon going to pair up with that codon. Let's bring it in. Oh, that little blue ball. That little blue ball is the amino acid. Okay, it's going to land in the ta-da, amino acid site, right? It docks. As it docks here at the P site, right, where we form that peptide bond, it's going to cause this amino acid that's attached right here. It's going to help to hook it up right there. And there she blows. Okay, we have made a little peptide bond between these two adjacent amino acids. This amino acid is going to, or this, I'm sorry, tRNA molecule is going to go over here. That's the E site, and bing, spits that sucker out. Bing, gone. This guy moves over into the P site. Oh, there it is. And now the amino acid site, the A site, is ready for new. Docks there. Peptide bond, yada, yada, yada. This continues until we've gone down from methionine, the start codon, all the way to the stop codon, of which we have 3. Once it gets to the stop codon, this whole thing disassociates, and ta-da, there's my polypeptide sequence, which is to say this is my protein. Just like we have a beginning, an elongation, and an end, we got the same thing here. We have an initiation for translation. We have an elongation phase where we're stacking amino acids in, connecting them, peptide bond, yada, yada, yada. And then we have a stop codon, right? And that's the terminator phase, the termination phase. So we have the same thing happening in transcription and translation. And if you really care about it, you can read the book. The book goes ad nauseum into it. Try to make it as streamlined as possible for us. Okay. But initiation, components necessary to start. I mean, small and the large subunit have to come together. Okay, yeah. I need the mRNA molecule. Yeah. I need the initiator tRNA. My start codon. Okay. AUG is the codon, and the anticodon is UAC. Small messenger RNA, initiator tRNA. It has the amino acid methionine, sorry. Okay, that's what starts this whole sucker off. EPA. And now it just continues like I said. Okay, we elongate it. What does that mean? We just bring in the tRNA molecule, the codon gets read by the anticodon with my specific amino acid, form the peptide bond. This one's going to go here. There it is. Hold on. Oh, see, it gets kicked out. Boom, gone. Next one comes in. And then this continues and continues, right? So the elongation just means it gets longer. What does that mean? I'm reading the damn strand of mRNA. And I'm adding amino acids. And this amino acid sequence is getting longer and longer and longer, hence elongation. Then it has to stop, right? I need a stop codon. I'm not asking you to know all of the stop codon names. I'm asking you to know where the start is, methionine, AUG. Okay. But I'm not asking you the names of the specific stop codons. You just need to know that there are 3 of them. Once we reach the period to the sentence, we are done reading that segment. And we have made my protein.
is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by...
We have a start codon, right? We have a specific spot where we start. Did we start at the very, 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 very tippy end where I put my little cap? No, I just said that this is the head and you should start reading down in this direction, down here. And then you're going to read towards the poly A tail. All right. So even though oh my God, it's okay. Okay. It's okay. It's looking for the start. You can't start, you don't know where the sentence is until you see the capital word, the capital letter, the word.
By a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the How is the information ribosome or RNA type of codon pairing occurs between the...
And I know I asked a few, I don't know how many. But I know I've asked some questions on the exam regarding, AUG is the codon, what's the anticodon? Okay, I know I asked that. So understanding Chargaff's rules is going to be something that you're going to potentially, put to action.
Codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
Once I've got my amino acid sequence, I've got my protein. Okay. Sometimes it'll stay right within the cell and do potentially something inside the cell. We can exocytose it. We can put it onto the plasma membrane, right? Through exocytosis. Still doing stuff for the cell. It could be something that we release into the fluids of the body. It's something that's going to be used elsewhere, not specifically in that cell. Sometimes we have to manipulate it just a smidge more, and we can add a sugar, we can add a glucose molecule to it, we can add a phosphate to it, we can add some lipids to it. I mean, we can manipulate this thing to be whatever it is the signals tell it to become. And it does that through chemical messengers in the body. And if we start talking about that, my head will explode. Okay. So there are just a number of glycoprotein factors in the body that say, hey, start making that one thing. Yeah, start making that amino acid, please. And then it'll start with our transcription within the nucleus. Copy it all. Unzip, unwind, unzip, expose, copy. I got my pre-mRNA transcript. Now I got to put a cap on the 5' end, a poly A tail on the back end. And I have to cut out the introns with spliceosomes, cut out the stuff that doesn't code, splice them back together, go throughout the nuclear pore of that nuclear membrane, go into the cytoplasm. This is where translation now begins. The mRNA molecule goes to the ribosomal subunits. We got our start codon, methionine. That says this is where you're going to start. This is the beginning of the sentence that I need expressed. Amino acids get brought in by the anticodon found on the tRNA molecule, bringing the specific amino acid. Docks into the A site. At the P site, form that peptide bond with that previous amino acid. E kicks it out, move on down the line. Next one comes in, and now I'm elongating this amino acid sequence. I'm making a polypeptide. Get to the stop codon, boom, ends the process. The polypeptide goes out. This is my protein. And depending upon all the other signals, it could be used right away as protein within that cell or in the cell membrane. Or I need to maybe manipulate it just a little bit more. So I'll take it to SmoothER, or I'll take it to something else, and I will, or the Golgi body, right? And just manipulate it just a little bit more where I add a couple more things to it, and la la. This protein, this glycoprotein, this glycolipid, okay, it can do whatever in the world I want it to do because chemicals in the body told it to become that. And that's how you make protein. That's how you make all the stuff that you see in the mirror every single day. That's how all that happens. Okay. Fascinating, interesting when it comes to this stuff.